Topic 12D, test crosses. A test cross is a genetic cross that allows one to determine the genotype of an individual. As an example, let's say we have a pea plant of unknown origin with purple flowers. We already know purple is dominant to white flowers, but we don't know the genotype for this individual. It could be homozygous dominant or heterozygous, and it would still appear the same. In a test cross, this individual would be mated to one who is recessive for the trait of interest. So in this example, we will mate the purple flowered plant to a recessive white flowered. The advantage of using the recessive white flowered plant is we know that it's homozygous recessive. So since we know this, we can begin to make predictions. And we can say if the purple flowered plant is homozygous dominant, then we know it will give a dominant allele every single time. The homozygous recessive, we already know, will give a recessive allele every time. So in this scenario, the genotype of all of the offspring will be heterozygous and will have a dominant or purple phenotype. In the second scenario, if the purple flowered plant is heterozygous and we then use a Punnett square to predict the possible genotypes, we notice that half the offspring will have a heterozygous genotype and a dominant phenotype, and half will be homozygous recessive for the genotype and recessive for the phenotype. To reiterate, if we set up a test cross with a fully dominant allele, where Mendel's law of segregation is in effect, then we would predict one of two possible outcomes. In one, half the progeny have a dominant phenotype and half have the recessive. From this, we can deduce the genotypes of the two dominant phenotypes in this cross. In the other scenario, all the offspring have a dominant phenotype, and from that, we can deduce that the parent with the dominant phenotype had a homozygous dominant genotype, and therefore all the offspring would be heterozygous and would appear dominant. To review, a test cross can be used to determine the genotype of an individual and/or inheritance pattern of a trait. In our example, we used a trait with a simple dominant allele and recessive allele that followed Mendel's law of segregation, and there were two possible outcomes. Each pattern informed us of a specific genotype.